Welcome to the Biochemistry Clinic. Today we are looking at ketogenesis, the production of ketone bodies. And these ketone bodies are acetone, acetoacetate, and hydroxybutyrate. So this usually happens when beta oxidation is happening at a very high rate in the liver. Now, when beta oxidation takes place, the presence of oxaloacetate is very critical because oxaloacetate is what combines with acetyl-CoA in the TCA cycle. But when there is no oxaloacetate, it becomes very difficult to utilize the acetyl-CoA that is produced from beta oxidation. Now, even if the ketone bodies are actually produced in the liver, they are not actually utilized within the liver. Why? Because the liver cells lack a very critical transferase enzyme that is required for utilization of ketone bodies. Now, the next aspect that I want you to know about these ketone bodies is that acetone is produced in smaller quantities, and usually acetone is actually exhaled, is given out. It's not among the ketone bodies that are actually utilized for energy. So that leaves us with acetoacetate and hydroxybutyrate. And these, like I said, have to be transported out of the liver to the extrahepatic tissues where they can be utilized, because in the liver they cannot be utilized. And this utilization actually requires these two ketone bodies to be converted to acetyl-CoA, which is oxidized in the TCA cycle. So hydroxybutyrate is actually converted back to acetoacetate, and it's this acetoacetate that is cleaved to produce acetyl-CoA, which can be utilized. So you notice that when you are starving, even the brain actually will adapt to make use of these ketone bodies, especially where glucose is not available. Now, let us quickly look at the steps of ketogenesis, or the production of ketone bodies. Like I said, this is happening because there is excessive beta oxidation in the liver cells. And what is beta oxidation producing? Acetyl-CoA. And these are our starting molecules, the acetyl-CoA molecules that are being produced from beta oxidation. And these acetyl-CoA molecules cannot literally go into the TCA cycle because usually during this period of starvation there is limited oxaloacetate because it is being used in gluconeogenesis. So you find that the acetyl-CoA that is produced in the liver at this particular time may not have sufficient oxaloacetate to actually combine with in the TCA cycle. Therefore, acetyl-CoA will now be converted to ketone bodies. And then when the ketone bodies are produced, they're actually moved out of the liver to the extrahepatic tissues for utilization. Now, the first step is a reversible reaction which requires the action of the thiolase enzyme, where two acetyl-CoA molecules are joined to produce acetoacetyl-CoA. Then, in the next step, we have HMG-CoA synthase, which actually adds a third acetyl-CoA molecule to acetoacetyl-CoA to form hydroxymethyl-glutaryl-CoA, or HMG-CoA. So you have two acetyl-CoA molecules to produce this, then you bring another acetyl-CoA molecule, add it to this, you're going to produce HMG-CoA. And then in the next step, HMG-CoA lyase, which simply means you split or you remove one acetyl-CoA molecule here to produce acetoacetate, which is a ketone body. And then from acetoacetate, you can actually generate your acetone via acetoacetate decarboxylase reaction. So you remove one carbon atom from acetoacetate, you produce this ketone body, which is acetone. And then there's a reversible reaction which makes use of NADH here. With the help of the enzyme hydroxybutyrate dehydrogenase, you produce hydroxybutyrate. Now, how are ketone bodies utilized? Like I said, it's hydroxybutyrate and acetoacetate that are actually being utilized. So you're going to notice that the utilization involves acetoacetate simply being broken down to two acetyl-CoA molecules. So in the first step, this is one of the critical enzymes that is very much needed in the utilization of ketone bodies. So acetoacetate is converted to acetoacetyl-CoA, which is then converted to the two acetyl-CoA molecules. So this acetyl-CoA molecule is actually, these molecules are the ones that are actually going to be used or utilized in the TCA cycle. So thank you very much for watching, and do not miss the next lecture.